replied to you that I will tell you what are the eubacteria and archaebacteria. I remember. Fine. So I told you all the characteristic nutrition of the bacteria, what type of the bacteria are there. Fine. So basically, let me remind you again that this is the kingdom Monera, and in kingdom Monera, we discussed that this is the kingdom. This is the kingdom where we study the bacteria, which are the basically uh, uh, where they are the basically bacteria, right? So bacteria have certain characteristic, and we have discussed all those characteristic, and they are ubiquitous, mean they are found in everywhere, right? Now we are going to discuss that there are two kind of the bacteria, means prokaryotes. There are two kind of the prokaryote, right? So let me discuss with you first. So in Monera, means Monera is representative of bacteria, but there are two kind of the bacteria. What are those kind of the bacteria? Eubacteria and archaea. Certain scientists have their few scientists have their views that bacteria should be divided into two groups. So let them divide into two groups. Okay. So basically, the scientists have divided bacteria. So bacteria means Monera. Scientist says that bacteria should be of two types. Why? There are two kind of the bacteria. Archaebacteria. You must have listened archaeological or archaeology department. So archae is itself, archae means old bacteria, right? So archaebacteria means ancient bacteria. Ancient bacteria. Basically, yes, that is true. These are the cells which first arrive on the earth. Normally, we consider that bacteria are first in the world, but out of the bacteria, archaebacteria are the first arrival in this world. Then another one there is eubacteria. And I have already told you that eu word means archae means ancient, not old. Ancient means before older one, and eu means new, right? So these eubacteria are modern bacteria, right? Modern bacteria. So what is with the archaebacteria? Archaebacteria are the most probably they are the first cell of the earth, right? Archaebacteria are characterized by very important. What is the peculiarity with the archaebacteria? I'll tell you what are the archaebacteria and how does they look like. So eubacteria we have already discussed. Eubacteria means, are you getting my point? Eubacteria means this bacteria, what we discussed. Now we need to discuss with the archaebacteria. Let me discuss it separately. What are the archaebacteria? Let me discuss in the next speech. So archaebacteria. Basically, this is the diagram of archaebacteria, right? Archaebacteria are normal kind of the bacteria. They look like normal bacteria, but they are different. What is the difference between the archaebacteria and normal bacteria, eubacteria, right? If you get any query, if you have any doubt, even small doubt, feel free to ask me, right? Never hesitate. So archaebacteria, so again, ribosomes are present. We know that the size of the ribosome is 70s in bacteria, and here also 70. Mesosomes are present. Mesosomes, they are like mitochondria, means they produces energy like mitochondria. They function like mitochondria. They produce energy. Plasma membrane is present, cell wall is present, capsule is present, DNA plasmid is present, flagella is present, membrane is everything which is present in bacteria. But this, then what is that? What is make special, right? So this archaebacteria, they are characterized by absence of, there is a absence of peptidoglycan in their wall. Peptidoglycan is not found in their wall, which is found in the eubacteria. Absence of peptidoglycan in their cell wall. In their cell wall, which is present in eubacteria, right? So first thing, and instead of this one, this contain a polysaccharide, right? Instead, their walls are, their cell wall are made up of, are made up of a polysaccharide, pseudomurine. Polysaccharide means sugar. Pseudo MUREIN, pseudomurine, right? Pseudomurine is a type of complex carbohydrate, right? And it makes them different. Now let me know one thing. Archaebacteria are found where or those point where the life cannot exist. They are found in extreme environmental condition where no cell can exist. Believe on me, virus do not have capability to stand in front of the bacteria. Virus are not that much strong. Why? Because a virus can be killed at 100 degrees centigrade. For example, suppose that you know that a particular food substance have virus, right? A virus is, it is infected with the virus. Heat up this substance up beyond the 100 degrees centigrade up to 110 degrees centigrade, all virus will die, right? And you can eat that thing. You will not get sick, right? But bacteria cannot be killed up to 100 degrees centigrade, right? They can survive beyond 100 degrees centigrade, right? Virus cannot be found everywhere, but bacteria are found everywhere. So this is the peculiarity with the bacteria. The same thing with the archaebacteria. Archaebacteria, they are found, they are found in extreme environmental condition. Environmental condition where no other organism can exist, right? So this is the best thing with the archaebacteria, right? Now the archaebacteria can be divided into three categories. They can further we classify archaebacteria. So archaebacteria can be classified into three categories. What are those three categories? You need to remember all those three categories. First category is methanogen. Methanogen are archaebacteria. These archaebacteria, they are found in marshy area. Found in marshy area, right? Where they produces methane gas. And methane gas is very harmful, very poisonous gas. No organism can exist in the presence of methane gas. But yes, archaebacteria themselves produces the and one more important thing, they are also found in the gut of in the gut of cattle. In the gut of cattle. Next, B. Another one, they are called halophiles, right? Halo means salt. Methano means methane. Halo means salt. Halophiles. Right. So halophiles, they occur in one more important thing. I want to ask you. I have a question with both of you. You must be aware with the Dead Sea.
Yes.
So what is the peculiarity of Dead Sea? Tell me. Which country do you belong to, Sidika?
Sorry.
From which country? You are
India.
from. You are from
Saudi.
India. Okay, okay. Martin, you are also from India. I thought you are from.
Yes, yeah.
Okay, I thought you are from Middle East. So tell me, what about the Dead Sea? What is the peculiar? Why we call it Dead Sea? That is a simple question. Both of you. Why we call it Dead Sea?
I don't know, sir.
Martin, what's about you? Can you tell me?
Not sure. I don't know.
Look, we call it Dead Sea because this is the only water body where there is no life. No life means you go to the any water body, you will find some plants, some algae, some small molecules, some animals, mosquito larva, anywhere. Even if you will analyze with the water which is flowing in your this sewer system, you will find there are certain animals and plants are present over there. But Dead Sea have that much salt concentration that the any cell cannot exist over. So no life exists over there. That's why we call it Dead Sea. You got density. This is the saltiest water in the world. But I have told you that bacteria are next to the God. Bacteria also, these archaebacteria exist there. So halophile bacteria are found in Dead Sea. No other life, no fish, no animal, no plant, nothing exists in that sea, Dead Sea. That much salt is there, but these bacteria are found. Right. So these bacteria are found, they are found in the high salt concentration. They are found in salt lake, salt lake, right? And they can bear high salt concentration. Right? High salt concentration, they can bear the high salt concentration. Okay, so they are the halophiles. And then third category of bacteria, they are called thermoacidophiles. Thermoacidophiles. So thermo means heat, acido means acidic, and philes mean this bacteria dwellers. Right. So these bacteria, from the name itself, it reflects they are found in the high temperature. Right. So these bacteria, they are found in high temperature, and they can bear high acid concentration as well. Acid concentration as well. So they can bear very high concentration, right? High concentration. I'm sorry. They can bear very high concentration of acid as well. Very high concentration of acid, right? So that's why they are called thermoacidophiles, right? So where they are found? They are found in thermal vent. What is the thermal vent? Thermal vent is basically the mouth of volcano which is found at the sea floor, right? They are called thermal vent. So they can the temperature at the thermal vent is 200 degrees centigrade. but these bacteria can exist over there, thermoacidophile bacteria. So these are the archaebacteria. They are the oldest bacteria, or they are rather than they are the oldest organism of the earth. So, and these archaebacteria are also known as the living fossils. These archaebacteria, they are also called living fossils. Is that clear, both of you?
Yes, sir.
Okay.
Yes, sir.
So this was the about archaebacteria, and one more thing which I want to add on. Give me 1 second. Charles Woos was, I think, the Charles Woos was the person who suggested the 6 kingdom classification system. Yes, that's true. Right. Now there is a person. Now you was talking, or you have asked me stated that what is the 6 kingdom classification system, right? So now again I came this PPT in the 5 kingdom classification system, right? So I will now you will come to know, because now I have told you. So here, what there was a person, his name was Carl Woos, right? So we know that bacteria comes under the Monera. So Carl Woos was a scientist. This is the proposed system, right? So let me tell you. According to in 1990. So I have told you that this 5 kingdom classification system was given by R.H. Whitaker in 1969. In 1990, there was a person, his name was Carl Woos. Carl Woos. Carl Woos said that Monera should be divided into 2 kingdoms, because what 2 kingdom? One should be Monera. In Monera, there should be only Ubek, right? And there should be a 6th kingdom. Okay? That 6th kingdom should be Archaea. And in Archaea, there should be Archaebacteria only. Archaebacteria. They should be Archaebacteria should be removed from Monera. Removed from Monera. And should be placed over here, right? So that the 6th kingdom was Archaea. But this is the proposed system, and there is a no 6th kingdom classification system. It was proposed by the, so let me write it down. Proposed 6th kingdom classification. Proposed 6th kingdom classification system. Is that clear, Shidika? Got it?
Yes, sir.
What is the
Yes,
6th?
sir.
This is the proposed system. Remember the name of that person. Normally, the name of the scientist and what is the 6th, that kind of the question, it is important for a medical examination actually. Okay.
What's his last name? Carl? And then?
Carl Woos. Woos. WOEST.
Okay.
Carl Woos.
Okay.
So, kingdom Monera. So archaebacteria, I told you, eubacteria. One more important thing I need to tell you. Okay, mycoplasma, and it is given in very short heading. Eubacteria, I told you. I need to tell you 2 more things. That what are the blue-green algae? Okay, I will add on what are the blue-green algae. Archaebacteria, I explained you, and 2 more. Mycoplasma and blue-green algae. BGA. So BGA stands for blue-green. They are a type of eubacteria. Blue green algae. The diagram which is given in your NCRT, that is of in bacteria, this nostoc bacteria. Basically, this is in eubacteria. You can see in the page number 19. The diagram which is given, that the diagram of nostoc. algae, blue-green algae, and mycoplasma. And mycoplasma. Okay. So what is the mycoplasma? What is the blue-green algae? This is very important. They all come under same thing, same umbrella. That is the Monera. So let's discuss. First, I will tell you about the blue-green algae. Blue-green algae are also known as the cyanobacteria, and they are basically eubacteria only. But there is a peculiarity with the blue-green algae. Blue-green algae are a bacteria which is capable of nitrogen fixation. Nostoc diagram, remember, this is important for your school examination. They may ask you to draw this diagram, so that's why you remember this diagram. This is Nostoc. Nostoc is a type of blue-green algae, BGA, right? So first important thing that BGA are blue-green algae are also called cyanobacteria. Cyanobacteria. Now we will talk about the nostoc. Nostoc is bacteria can never be multicellular. So what we will call it? Tell me. Martin, tell me. Shidika, tell me. I was telling you that bacteria can never be multicellular, but there are a number of the cells.
Some bacteria can only be.
Yes, Martin.
Some bacteria only.
Some bacteria will be multicellular. Exceptionally.
Yeah.
No. Bacteria can never be multicellular. Still, I stick with my statement. Bacteria can never be multicellular. So what is that? This is just a collection of cells. Right? Why? Because these cells do not have their protoplasm is not interconnected. Until the protoplasm is not interconnected, you cannot call it multicellular. There is no interconnection of the protoplasm. They are just bind with a substance that is called mucilaginous sheath. Mucilaginous sheath. It is made up of the carbohydrate, means sugar, and it is gel-like substance. So they are bind with mucilaginous sheath. So what we will call it, right? The collection of bacteria is called colony of bacteria. What we will call it? Colony, right? Colony of bacteria. This is a colony of bacteria. Bacteria can never be multicellular. Why? Because there is no protoplasmic interconnection. As there is no protoplasmic interconnection. Are you able to read my handwriting? Is it legible, or should I write more clearly? Is it legible for both of you?
Could you write it a bit clearly?
Should I write it more clearly?
Yes, please.
Okay. Yeah, please tell me whenever you feel that this is like this way. I was thinking that you will get it. Okay, fine. So let me write it down more clearly. BGA is also called cyanobacteria. Is it clear? Clear now?
Yes, sir.
Okay.
Yes.
Okay, fine. And I know where you will not get it. We call it colony of this collection of bacteria is called colony of bacteria, as there is no protoplasmic interconnection so we can't call it multicellular. Right? So this is like this way. Fine. So there are particular kind of the cells. So nostoc have particular kind of the cell. These cells are called heterocyst. What is the function? Nitrogen fixation is the function of heterocyst cell. Right? Fine. So heterocyst cell of nostoc fix nitrogen from air into soil. Clear? So that they fix
Yes, sir.
soil, they fix atmospheric nitrogen into soil nitrogen. That is their function. So this is the peculiarity of the blue-green algae. Let me see if there are some more peculiarity of blue-green algae. They are also called cyanobacteria and okay, heterocyst. I told you. The rest of the functions are same as the mycobacteria. And one more very important. Okay, so these bacteria, one more important thing, they have these blue-green algae, BGA, also have chromatophore. What are the chromatophore? Chloroplast is not found, right? So they have chromatophore. So chromatophore, they are meant for photosynthesis. So with the help of chromatophore, as chloroplast as chloroplast are not found here, right? So chloroplast are not found. That's why because chloroplast is not present over there. Then in place of the chloroplast, chromatophore. You have to remember chromatophore are found. And what is the function of chromatophore? Chromatophore are meant for the photosynthesis. Is that clear? Tell me, is it clear?
Yes, sir.
Very good. Note it down. All this point. Tell me when you note down the points. Chromatophore very important.
Sir,
Yes.
what is the difference between a nucleoid and a nucleus?
Nucleus, actually, nucleus is a qualification. To get this qualification, you need three things: nucleoplasm, chromosome, and that is
So
surrounded
it's
by nuclear
written
membrane.
here, coli and C bacteria is called. The second point, what's it?
Second point, this one, collection of bacteria is called colony of bacteria,
Okay, thank you.
as there is no protoplasmic interconnection, right? So, Shidika, look, here the bacteria have chromatin or chromosome. Bacteria do not have nucleoplasm. Bacteria do not have nuclear membrane. So two things are missing. So that's why they do not qualify the term nucleus, right? So we do not use nucleus term. To get a nucleus term or to get the qualification of nucleus, you need three things: chromatin fiber, uh, nucleoplasm, and nuclear membrane. Rest of the two things are missing over here. So we do not use nucleus term. We call it nucleoid. Clear?
Okay, sir. Yes.
Yeah.
So what's the third point?
Third point, this one, heterocyst cell of nostoc fix the nitrogen from air to soil.
Done, sir.
Done. Martin, done.
Good answer.
Okay. So last but not the least, that is the come under the Monera. So that is the mycoplasma. So let me discuss here only in this case. So mycoplasma is the smallest organism of the world, right? So mycoplasma. Mycoplasma. Mycoplasma is also known as PPLO. What is the full form of PPLO? PPLO. PPLO is pleuro pneumonia like organism, right? Pleuropneumonia like organism, PPLO. So actually, first name was PPLO, right? So basically, these organism was not neither like bacteria nor neither like eubacteria, nor like blue green algae, nor like archaebacteria. So scientists were confused what they are. They look like something the bacteria or something organism which spread the pneumonia, right? So that's why they given the name PPLO. Then when they came to know their feature, their function, they have said that they are a different organism and they given the name mycoplasma. So nowadays we know them with the name of the mycoplasma. But mycoplasma and the PPLO both are the same thing, right? So try to understand. Oh my, it's a very big diagram. Make it a smaller one. So this is the mycoplasma. It's a diagram of mycoplasma. That is the bad thing with the NCRT. They do not discuss everything. They do not mention everything in detail. So this is mycoplasma. Look, mycoplasma, as I have told you, that this is the this is different from the bacteria, blue, but this come under the Monera only. Right? We keep them under the Monera. So look, they are lipoprotein membrane. So they are surrounded by the single membrane. Lipoprotein membrane means they are surrounded by the biomembrane or plasma biomembrane. Right? To either call it biomembrane or plasma. They have some soluble RNA, so RNA are present over there. They have some DNA. Ribosome is found. Ribosome size is same like the bacteria. The size of the ribosome is also here 70s, and there are some soluble protein, right? And inside there is a protoplasm. Protoplasm is present over there, right? And then at the end of end of this DNA, right, there is a disc-like structure. This disc is called replication disc. From here, the DNA replication disc. So tell me, what type of the DNA bacteria have? We have the straight DNA, but bacterial DNA is circular. Remember, bacteria have single circular DNA.
Yes.
Okay, but here the DNA is not circular. So that here they differ from the bacteria. So mycoplasma, they are also called PPLO. They are also called molecule. They are also called molecule. MOLLIQUTES. UTES, molecules, and they are the they are the they are simplest and smallest free living organism. Simplest and the smallest free living organism of world. Organism of world, right? They are the simplest and smallest free living organism of the world. Uh, their size is their size varies from 0.1 to 0.15 micrometer. Next thing, so plasma membrane, cell wall is found in bacteria. Cell wall is not found. Is it legible right now or not? Is it legible now?
Yes, sir.
Cell wall
Yes,
is
sir.
not present there. They do not have cell wall, right? Cell wall is not found. So bacteria have cell wall. Their DNA, they have, they have straight DNA. Straight DNA. While bacteria have, while bacteria have circular DNA. Circular DNA. So their DNA is straight DNA, but bacterial DNA is circular DNA. That they also, and they can change their shape. They can change their shape. So what we call them, there is a particular term for those organisms which can change their shape. That's why they are called pleomorphic, right? Pleomorphic, which can change their shape. Pleomorphic, right? That's why we call them pleomorphic, because they can change their shape. Their size, I have told you, they are the smallest organism, and the rest of the thing is matches with the one last thing. Majority of them are parasitic. Majority of them means they spread disease are parasitic. Note down, please. Let me know when you note it down. Mycoplasma, protestant,
Done, sir.
Done. Shizo?
I'm doing so.
Okay. Yeah, please tell me when you do.
Sir,
Yes.
what's the difference between straight DNA and a circular DNA?
Straight DNA, that both end are open, right? Like it's like thread, right? If you have two thread and you have, it's like you must have seen that wire, which is usually an electrical wire. One wire is red, one wire is blue, or one wire
Yes.
is red and black, right? You must have seen that wire. The DNA,
Yes.
rDNA is like similar like that. That kind of straight DNA, that is straight wire. So when this wire will replicate, it will open completely, right? And it will become twice, right? From 1 to 2, 2 to 4.
Yes.
That's how DNA replicate. But bacterial DNA is like circular, like ring, right? Like ring. It will replicate. When it will replicate from one ring, another ring formation will take place, right? And the
Yes.
circular DNA is not very a big DNA. It's a very small DNA. But the straight DNA can be, in our nucleus, there can be 1 kilometer DNA in one nucleus,
Ah,
right? So
okay,
that is the
sir.
beauty of circular DNA cannot be bigger one. One more important thing. Gradually, I will tell you. So right now, I am trying to be a very simplified, right? So when you will come to like you will get adapted with like my pattern, then I'll give you much more information. Look, our DNA is made up of two things.
Yeah, done, sir.
DNA is so our DNA, like we have chromosomes. Chromosomes are made up of two things: histone protein plus DNA. Okay.
Yes.
This is a histone protein, and I wrap up like a thread around this one. So this will become chromosome, right? So our DNA is made up of, our chromosome is made up of two things: histone protein and DNA. But bacterial chromosome do not have histone proteins. That is also a difference. Okay.
Okay, sir.
Fine. So look, here we have studied one thing, that kingdom Monera. It is Monera have three representative organisms. What? So in Monera we have incorporated three things. So Monera is, we know that Monera is kingdom of prokaryote, right? That agree?
Yes.
Kingdom of prokaryote. And here we have studied three kind of the organism. First, eubacteria. And eubacteria, we have studied general eubacteria. Then along with that, we have studied blue-green algae, BGA, right? Second one, mycoplasma, right? So we have studied mycoplasma. Third one, archaebacteria. So these three are a representative organism of kingdom Monera. Clear? Agree with that?
Yes, sir.
Now come to the Protista. Now we will start with this next kingdom, that is the Protista. Today I will introduce, and next class we will start with the Protista. So the second kingdom is kingdom Protista, right? So Protista, here we will study one thing. Again, if we talk how did the Protista kingdom separated, so we have discussed in detail that this is the kingdom of eukaryotic unicellular organism. Agree? This is the
Yes.
kingdom of eukaryotic, means they will have nucleus, and unicellular organism, right? And further we divide it into So kingdom Protista have been divided into this category. Kingdom Protista have been further divided. How many types? So first, they are the photosynthetic protist. Photosynthetic Protista. Those Protista which can make their own food, they are called photosynthetic protista. Who will be part of this one? Example, there will be, we will include them algae. So algae will be part of them, right? The second category of the protista, so they all are unicellular. So here the plant group will be kept over here, right? So we have taken out the unicellular photosynthetic organism from the plant and kept in the protista. That's why protista is called mixed kingdom, right? So from where we have taken out, we have taken from the plantae. Unicellular plant have been taken over here in protista. Clear? The second one, consumer decomposer. Consumer decomposer protist. Who are decomposer? Here the like slime mold. Organism is slime mold. Slime mold have been brought from fungi. So from fungi, unicellular fungi have been taken out. We kept them in protista. So first category taken out from algae, sorry, from plant. Second category taken from fungi, and third protozoans. Protozoans protista. So protozoan protista, they are like amoeba. They have been taken from animals. So we have taken out all the organism. So from where? We have taken off from plant, from plantae, we have taken this part. From fungi, we have taken this part. And from animalia, we have taken this part. So we have taken out unicellular eukaryotic organism from every kingdom and make a separate kingdom, that is protista kingdom. That's why protista is called mixed kingdom. Is that clear?
Yes, sir.
It is called mixed kingdom.
Sir, what's the example for the protozoan?
We will discuss in detail. We will go inside the photosynthetic protista, consumer decomposer, everything, right?
Okay.
So this is just I'm telling you how does the protista kingdom have been made, right? And then now we will start what is the protista, what a protista exactly is, right? So example of any protista. Protista. So this will be Monera. That part we will start with the next class, right? Protista part, right? So go through with it, whatever I am saying, whatever I have taught you today, and ask me if you have any doubt. Protista, the representative protista can be amoeba. That is also representative of protista. The best representative of protista. Some algae, right? Like I'll tell you in detail in next class. Then you will come to know. Tell me, everything clear or noted down? Martin and Shidika.
Yes, sir. Everything is clear.
I'm
Very
noting down, sir.
Okay. So are you studying after the class as well? And with your
Yes.
NCRT with reference book, right? Keep
Yes.
on studying. Keep on studying. So right now, in starting, I will keep my pace slow, so that The best thing is that after 5 or 10 class, 5 or 6 class, not 10 class, 5 or 6 class, you will synchronize with my frequency. I will synchronize with your frequency, and our pace will become faster. Got it? And one more very important thing I want to tell you. Just we have completed our crash course for like since number of the years, like at almost from the 10 year. Whatever the date of the completion of course, I will commit with you, right? Till that date, I will revise your syllabus at least for the 4th time. That is for sure. That is my commitment, and I always adhere with that time, right? So never have that kind of the doubt that will it be completed on time? Not, but that is not an even question, right? Suppose that I say that I'll complete your syllabus on the December 15. So believe on me, December 15 is not the syllabus completion date. I will complete it on the October itself, and I will revise 4 times before your examination. Practice and all.
Sir, what is the example? What's the example? Third one's example at protozoa.
Protozoa, we will discuss one by one. It's very detailed. It's
No,
very
no, no. What is written there? What is written there?
protozoa. Okay,